Hello everyone, my name is Eric Stegmann, and I will be presenting over my thesis work that was completed in 2020 over the habitat selection and host detection in the salamander mussel Simpsonaias ambigua. Freshwater mussel reproduction is a delicate method of parasitism that requires a multitude of factors to align to be successful. Adult mussels produce larvae called glycidia in a variety of different manners. These glycidia must then attach to a vertebrate host, usually always a fish, and often will only be successful on a single species of fish. Once attached, the glycidia will transform over a period of time until they are juvenile mussels, which will drop from the host and sink to the substrate to burrow and hopefully begin their journey towards adulthood. Mussels generally must be able to detect the presence of the host and/or attract their mobile host to come to them. Fish hosts are often visually drawn to mussels due to mussels developing tissues that look like potential prey, often done through the packaging of glycidia and conglutinates, or altering mantle tissue that looks like insect or smaller fish. The mussels with these mantle lures are sometimes responsive to their host and are able to move the lure when a fish swims by, causing vibrations in the water or casting a shadow over the mussel. There have been some studies investigating other methods in which mussels may be able to sense their host other than physical disturbance. When restricting physical access of host fish to brooding female mussels, some females were still releasing a fair amount of glycidia, potentially being able to sense the host fish through some kind of chemical signal. Glycidia are also known to respond to chemical changes in their environment by snapping shut when in the presence of mucus or blood. Like I mentioned before, almost every mussel species uses a fish as their primary host. However, there is a mussel species that breaks this trend, the salamander mussel, Simpsonaias ambigua. This mussel is relatively small, growing to about 2 to 5 centimeters long as adults, and they're fairly active. Seen in this video, they weren't recently agitated, and most of these individuals end up moving in a pretty short clip. Simpsonaias is often found under large flat rocks grouped with conspecifics, as well as its only known host, the mud puppy, Nectaris maculosus. Nectaris has been often found laying their eggs in rock crevices during the spring, and females get fairly territorial over these. This is an important timing for Simpsonaias to interact with its host and to ensure glycidia get to the gills. Due to the common co-occurrence of this mussel and host in the same habitat, I wanted to test if Simpsonaias was able to seek those types of areas as preferred habitat, those areas being dark, offering a flow refuge, as well as a structure to offer a physical barrier to movement. I also wanted to investigate if Simpsonaias may possibly be detecting its host through chemical cues and orienting their movement towards that host. Between these tests, I was hoping to see if there was a reason these species are often found together, other than possibly just juvenile deposit from the host in a limited stable habitat. To conduct this research, I began by traveling to the Genoa National Fish Hatchery located in Genoa, Wisconsin. Here, Simpsonaias and Nectaris were already being held for propagation, and thankfully the staff at Genoa were gracious enough to allow some of these animals to be used for testing. To assess habitat preference, miniature streams were constructed in plastic shoeboxes. Each box would be filled with a layer of sand for substrate. Water from a reservoir would be pumped to the upstream end and passed through a foam block to normalize flow. Flow was assessed by filling a beaker and timing how long it would take to fill 300 milliliters, and adjustments were made to the pumps to make sure that they were all flowing at the same rate. Water would fall back into a collection reservoir and be recycled, and after each test was done, half of the water from that container would be replaced to avoid ammonia buildup within the system. To offer artificial habitat, glass or tile were propped up with PVC pipe. 2 of each of these were placed in alternate positions both upstream and downstream. 10 mussels would be systematically placed throughout the box, 1 under each structure and 6 down the middle. After 24 hours, position would be assessed. There was an unexpected behavior observed, as 17 of the 300 total mussels tested were able to climb the wall and jump from the exit pipe into the collection bin below. Scent trials were conducted in two-choice flumes, where two independent streams were introduced upstream of an open choice area. Water would flow through several layers of screens and foam to normalize flow so that once it went through the open area, the two streams would not mix. Here I have an example of what that looks like, where I used red food coloring on the left-hand side to indicate that that stream was not mixing with the stream on the right-hand side in this open area. Water was not recycled in this system, as water that exited the system would fall to the drain on the floor. Water would be replaced in the reservoir every 30 minutes to try and keep the pumps under similar pressure throughout the trial. Water from either the well used as control, water from the tank holding mud puppies, or water from a tank holding bass would be pipetted into the upstream section every 2 minutes, and mussel location in the choice area would be recorded every 5 minutes for 2 hours. After each trial, the box would be drained and rinsed out. For each scent combination, a total of 30 trials were run. The final set of trials I ran was to investigate if situations like hypoxia that might be encountered in this limited habitat would affect Simpsonaias behavior. These tests were done at Missouri State, and the way this system worked was water would start out in this cooler reservoir and be pumped to the top of this oxygen stripping ladder. As it falls down, denoted by this red arrow, water would interact with nitrogen gas, thus stripping all the water of oxygen. This blue arrow indicates normoxic water, and these would run through the same two-choice flumes indicated as before. Animals were tested in a similar manner as the scent trials were. 30 tests were run between 14 and 16 degrees Celsius, and about 20 trials were run at between 20 and 22 degrees Celsius. For all results, error bars indicate plus or minus 2 standard error, and different letters will indicate statistically significant differences. Any red dotted lines will represent expectations under totally random events. So when looking at habitat boxes, there were significantly more mussels found in the upstream half of the box than downstream. On average, there was about 8 found in the upstream half compared to only 2 in the downstream. Due to the shelters only covering about a third of the total area in the habitat boxes, habitat selection ratio was used to compare mussels found on uncovered and covered substrate. Given the relative area, there were significantly more mussels found under any type of shelter compared to mussels found on substrate that was not covered. When looking at mussels that were only found under shelters, there were significantly more mussels found under a dark flooring tile compared to those of clear glass, indicating a selection to avoid excessive light. When looking at the number of mussels found along the edge of the box and/or shelters, again, the habitat selection ratio was used as edge was only about 20% of the total area in the box. However, significantly more mussels were found along the edge of that box, indicating that Simpsonaias might be moving until it finds that physical barrier to stop movement. The average proportion of mussels found within a body length of each other was much higher than expected. Expectations were made using a Monte Carlo simulation in which cells were denoted as either mussel or empty. Each run of the simulation would count how many mussels were in contact with one another. However, from the data collected, it is not clear if mussels are aggregated due to limited preferred resources or if there is a conspecific attraction motivating that movement. For scent trials, the average proportion of observation was used to compare the amount of time Simpsonaias spent in the presence of each scent type. Across all scent combinations, Simpsonaias did not spend a significant amount of time in one type over another. In hypoxia trials, there was no significant difference between the amount of time spent in hypoxic versus normoxic waters at cool temperatures. However, when the temperature increased, more time was spent on average on the hypoxic side. Mussels may have used all available oxygen and panicked and clammed up once on that side for too long and weren't able to move out for the rest of the trial. So in conclusion, whenever it comes to habitat choice, Simpsonaias appears to be choosing dark habitat that offers an edge. This may be active choice, or simply host deposition of juveniles, and that's the only stable substrate they have. Aggregation is not clear if it happens that they are seeking one another, or if they happen to just be finding that same limited habitat type. Simpsonaias was also recorded to have the ability to climb walls, and that reason is unclear. They may do this to try and avoid competition for space since they're found in such high densities. They may be trying to do this to avoid hypoxic conditions, and they may be trying to reposition themselves around their host. However, for the hypoxia, none of the climbing behavior was ever observed during hypoxia trials. Simpsonaias appears to not respond to chemical cues from mud puppies or fish. And Simpsonaias showed a stunned behavior when exposed to hypoxic waters, and this behavior became more prevalent whenever the water was at higher temperatures. Again, this may be because they used up all available oxygen in the water, and they were trying to preserve what oxygen they still had in their system. I want to thank the Missouri State Graduate College for funding this research, all of the members of my committee who helped with method development and results. I want to thank Brian Grindstaff, who was an excellent machinist at Missouri State who helped put all of this stuff together. A special thank you to Megan Bradley and Beth Glidewell from Genoa for allowing me to use all of these animals, and all of the other grad students and undergrads in the Barnhart Lab who were there along for the whole ride. And with that, I am happy to take any questions.